So we're moving to our last speaker, last but not least, Yenisleidy Díaz from Instituto de Biologia Molecular y Celular de Rosario, Argentina. Please, Yenisleidy, can you share your screen?
Thank you. Yes, of course.
So I'd like to ask you to stick to your time. We're a little bit, okay. Running out of time.
Okay. Yeah. Can you see my screen?
Yes.
Okay, perfect. Hello, everybody, and thank you to the organizer for giving me the opportunity to give you this talk. My name is Yenisleidy, and today I'm going to share some results obtained during my PhD thesis and carried out in departments of biological chemistry in Córdoba under the supervision of Maria Laura Fanani. Today, I'm going to talk about how miltefosine inhibits the membrane remodeling caused by phospholipase actions by changing membrane physical properties. Miltefosine, or hexadecine phosphocoline, HEPC, is a key phospholipid analog. As you can see in this structure, it's amphiphilic molecules with the single hydrocarbon chain and phosphocolines as a polar hair group. HEPC shows cone-like shape and was determined to belong to the type 1 lipid group, and favoring mesos and small lamellar structure. Originally, it was developed as anti-tumor agents in the late of 98, and however, further studies demonstrated that this amphiphil is active against most Leishmania species. Despite this mechanism of action is not completely elucidated and has been linked its activity mainly to interferes with phospholipid metabolism and affects the biosynthesis of phosphocolines, altering intracellular cholesterol traffic. Also affects proliferation and cell survival signaling pathway by inhibition of protein kinase C, apoptosis, and also with modulate some phospholipase activity. In all cases, the HEPC actions indirectly involve the interaction with biological membranes. For this, our first approach was to explore the interaction of HEPC with lipid membranes, and our works dealt with two important questions: HEPC membrane selectivity and its actions as a membrane perturbator agents. And for this, we use lipid monolayers or different rheological characteristics, and we combine penetration and superficial titration studies, as I show in this scheme. And we employ for this several commercial lipid compounds for the construction of the artificial membranes. And these lipid monolayers were composed of pure lipids that were previously characterized and being the different phase states, liquid expanded, liquid condensed, solids, or liquid order. We estimate, and we, for the penetration experiments, and we graph the P increase after the insertion of this amphiphilic into different sector membranes, and the results that I show in these blue bars. And in this case, you can see that we observe an increase in P increase than 10 millimeters per meter in all cases. And this generally observing a greater increase in more compact membranes that you see, for example, for DBPC, and compared to most expanded ones as CHISAS and POPC. And to estimate the amounts of that as this amphiphilic got incorporated into those membranes, this pressure, and we carry out supertitration experiments, and we determine the fractions of HEPC necessary to achieve a 60% of speed saturations, and the results I chose in red bars. In this, you can see that we obtained a greater incorporation of the amphiphilic in fluid membranes compared to most expanded ones. We also observe that these amphifields alters the film's property when incorporated into different monolayers. And thus, the experiment was evidenced by comparisons was the compressivity modulus parameters before in black bars and after the penetration process in gray bars. Our results clearly shows that the insertions of this amphiphily into different membranes decrease the compressivity modulus values, and this indicates that these amphifields have the ability to convert these films into more compressive monolayers in all cases. And from the other experiments from calorimetry using MGUVs, in that case of DPPC in black curve, in the presence of subconcentration of AGPC, we observe a decrease in transition temperature. That means or reflects a thermodynamically more favorable transition to LA phase in the presence of the drug, and it supports that a preferential partitioning of AGPC into fluid phases. So to sum up, our work proves that AGPC can integrate to lipid membrane, has a preferential partition into fluid phases, and its incorporation results in with this in mind, taking into account that membrane fusions are proposed as a target process in HGPC actions, now we were wondering how could changes in membrane properties induced by HGPC incorporation have the potential to affect some phospholipase activity. For this, to understand the regulations of HGPC on the actions of phospholipases on lipid membranes, and we use LGUVs as a model membrane with the composition contains the substrate of phospholipase C, a fingomyelin. The endose products of those enzymes are the acetylglycerol and ceramide, and those metabolites is well known. There are inverting cone-shaped lipids, and its effect has been reported that alters the property of the membranes and also induce restructuring of the membrane with aggregates with high curvature. And this evidence led us to formulate the hypothesis that due to stump-shaped HGPC incorporation into lipid membranes alters curvature or generate a curvature stress. And we set out the main goal for this study to understand the effects of HGPC on phospholipase activity, enzymes activity, and also how membrane physical property changes after the insertions of this kind of amphiphilate. We first analyzed the effects of HGPC on the enzyme activity from PLC and sphingomyelinase, and we determined the initial velocity and the results I show in this table. When liposomes were treated with PDC, both in the absence or in the presence of HGPC, reaction rates here remain similar, but in the case of the sphingomyelinase activity, we observe a little inhibition of the reaction rates by AGPC. And in the other hand, we explore the ability to HGPC to alter the high curvature membrane structure induced by lipids, acetylglycerol, and ceramide. That these metabolites lead to fusions and aggregation process. And for evaluate this indirectly, we do it through turbidimetric assays and also by changes in vesicle size distribution profile using DLS, dynamic light scattering. When liposomes were treated with the enzymes, responds to an increase in size of the lipid particles, increasing the scattering of light and their absorbance at 500 nanometers. As I show in this figure in black lines, when PDC is added to the LUVs, an increase in turbidimetry is observed when the acylglycerol productions. But when we added 20% more of AGPC to our system in blue lines, and clearly shows a drastic decrease in vesicle aggregation. For its sphingomyelinize, and we obtained a similar behavior. The addition of these enzymes, we observed an increase in turbidity in the first 5 minutes of the reactions, and in the absence of the AGPC, followed by the realization process. But in the presence of this amphiphilic, we observe that this process occurs in a slow way. As these results were confirmed by measuring of particle size by DLS, and I showed the representative graph of the different population size distribution of lipid particles before in black symbol, and after the PLC treatment in blue symbols. And we observed that the enzymes production of the acylglycerol by PLC induced the appearance of the populations of large particles. And in the presence of the AGPC, reduce this effect, and we observe here smaller vesicles. For both enzymes, and we obtained similar results. Also, in addition, we carry out quantitative analysis of the extent of the hydrolysis, and we quantify the amount of substrate produced for both enzymes at long times. And we found it that after 60 minutes of the enzymatic treatment, the acylglycerol reduced to a 4. So these experiments says that AGPC inhibits PDC activity when studied over longer periods, and also cause an inhibition of the vesicle aggregation. For both short terms and long terms, in the case of sphingomyelinase, AGPC induced up to more than 25% of inhibition of the amount of ceramide forms. And these results shows that AGPC affects sphingomyelinase dependent liposome aggregation to a greater extent than enzymatic activity. With these results, we further explore the effect of AGPC on fusogenic liposomes. For this, we use giant unilamellicar vesicle as the fusion target. So we did a Fred membrane assay. And this experiment was adapted from the work described for the MOBA groups in these systems, and we use HUVs labeled with donor probe generated by electroformations and designed as not substrate of phospholipase. And those HUVs were mixed with fusogenic LUVs with our substrate of the enzyme, and also were labeled with rhodamine in the presence or absence of the AGPC. And finally, we added the phospholipases. This vesicle fusion after the enzyme additions is detected by 2 means. One, lipid mixes increase threads between 2 probes. In that case, the red emissions of the acceptor probe increase at the expense of the emission of the donor ingredients upon the detention of the donor probe. And besides, we detect fusions, membrane fusions events through morphological changes of the HUVs observed by confocal microscopy upon the enrichment in the acylglycerol or ceramide. Here I show the result that we obtained when the HUVs mixed with SUVs with this composition without PLC were observed by confocal microscopy as a circle. And when the addition of the enzyme to this mixture triggered the acylglycerol formations in SUVs, and we observed changes in the SUV shape over the times, and with evidence the occurrence of membranes areas with higher curvature. And these fusion events also was detected by semi-quantitative analysis of the fluorescence intensity in radio and red-green in each condition. As I show in gray bars, there was an increase in red-green fluorescent radio. There is an increase in red signals, 30 minutes, that doubled the initial value here. So this means that we observed or we obtained fusions in the absence of fusion events in the absence of the amplifier. Remarkably, when we added to these systems 20% more of HEPC that was present and remembered in the LUVs, the fusion to the giant lipid vesicles was reduced. And those events reflected by this low value of red-greens and fluorescent radio, and also because we don't observe no shape change in SUVs over the time. And in the other hands, for the other enzymes, the sphingomyelinase and SUVs mixed with SUVs with these compositions were observed as a circle by confocal microscopy. And the addition of the enzyme to this system triggered the formation of ceramides in SUVs. And after the few minutes, we observed red dots that attach to the SUV membranes. In this stage, we interpreted that SUVs was associated but did not mix with the SUV lipid membranes. And also, as the enzyme reactions progress and shows SUVs shows increase in red fluorescence, that this means an increase in fusion events, until we observed at 30 minutes of the experiments that SUVs collapse in a lamellar lipid structure that we observed like this amorphous lipid cluster. Here I show a little video when we can see this process. Notably, the addition of HPC to this system clearly shows a reduce in the SUV destabilization after the sphingomyelinase and straights. Here I can see that we don't observe change in the shape of the giant vesicle membranes. To sum up, our works indicated that HPC does not affect the initial rate of the reaction catalyzed by PLC or the sphingomyelinase. However, the extent of the hydrolysis is inhibited by HPC over longer times. This results shows that HPC inhibits membrane restructuring events caused by the acylglycerol and ceramide production. And with this, those evidence, we propose a model represented by this cartoon, where HPC has the ability to modulate the action of phospholipase in a classical way, related with non-specific geometric compensation by altering of the intrinsic curvature of the membrane. HPC is as a single title alkyl phospholipid analog that have an effective cone shape and geometry, and when it inserts into bilayers, can induce a positive curvature and counteracting the effects caused by the acylglycerol or results suggest that the HPC may act in a classical way and may be influenced in not only those enzymes, maybe but a greater variety of membrane-bound enzymes sensitive to curvature stress. So finally, I would like to thanks to the institutions provide funds to carry out this work, to a great CQB biophysics group of Córdoba, PhD Dr. Carlos Mas and Cecilia San Pedro for technical assistance in confocal microscopy images and capture and analysis, and especially to Ernesto Ambrosio and my advisor, PhD advisor Maria Laura Fanani. And if you want to obtain further information about these results, you can find it here. Thank you all for your attention, and I look forward to your comments and questions.
Thank you, Yenisleidy. There is one quite quick question, so then we can wrap up our session. Is there any question from the audience? Don't think maybe there is no question. So if there is no question, I'd like to ask all the speakers to turn on their cameras so we can just make a closure of the session. Hope you're all there. So I'm not sure if Natalie is still there.
Yeah, I'm here. My video won't turn on.
Oh, Marcelo, can you help with that?
Yeah, I think that Marcelo has blocked all of us. I can't turn my video on either. Eduardo, just as we wait, let me just take this opportunity to remind everybody that the registration for the IUPAB meeting in October is now open. So please visit our website and register, and also help us advertising the event within your network of collaborators and colleagues. Okay, so it's very easy to find the website. Just type in on Google IUPAB 2020, and will be the first link to pop up. Okay, so registration is open, and I invite everybody to register.
Sorry, just to remind also that we have some fellowships available for students. So it's possible that you can participate in the meeting without paying anything. And if you take a look in the website, you can find the information, or you can email us and we can give you the directions to apply to the fellowship, and then you can participate for free in our meeting. Thank you.
So I hope Natalie can turn her camera now.
Yep.
Yes, she can. So with that, I'd like to ask for a round of applause for everyone that all of the great talks we had today, and thank the speakers for their time and effort to make these great presentations today. And I'll also I'm just showing our upcoming webinar in 2 weeks. So I also invite you to join us again if you are available. And yeah, have a great week and be safe. So thank you again. Thank you very much for your time and presentations.
Thank you.
Thanks, everybody. See you. Take care.
Bye.
Stay safe.
Take care.
Bye.
Thank you, guys. Congratulations to the speakers. Thank you.
Thank you.
Thanks.
Bye.
Thanks.
Ciao.
Congratulations on this nice work.